Hi, everyone. Welcome to this week's Exploration Club session. So for today's session, we'll be focusing on CD33 as a therapeutic target in acute myeloid leukemia. Okay, so to give a bit of background and sort of introduce this, so today we'll be specifically exploring sort of the potential of CD33 as a therapeutic target in AML, which is a type of blood cancer known for rapid accumulation of abnormal white blood cells. While treatment options for AML has improved over the years, many patients still face poor prognosis. So in this session, we'll mainly be focusing on CD33 and its significance in AML using different functionalities within the talk to data system, and particularly how we can target CD33, learn a bit more about CD33 co-targeting effects, and how can this sort of provide new pathways and information to have more effective treatments. Okay, so to really allow any new scientists who are not really used to working in this area, so to better understand also the significance of CD33 in the case of sort of acute myeloid leukemia, let's explore sort of the normal process of hemoptytosis. So in AML, the differentiation of hematopoietic stem cells is impaired, and this leads to the accumulation of immature myeloid blasts. The blasts show apparent activation of the signaling pathways, and the oncogenes prevent the normal differentiation of these cells. So if we look more into sort of AML, normally this sort of diagnosis is confirmed based on the following markers. So we would need to be able to detect myoblasts with MPO, CD13, CD33, and CD117 to be able to really provide the highest specificity for these myeloid lineages. And so this in terms of sort of the aspect of the different myoblasts, so the identification of over 20% of these myoblasts in the peripheral blood confirms AML. And the specific genetic transcriptomic markers like CD33 can help target this abnormal cell population. Okay. Okay, so to get a bit more information, why CD33 works also as a therapeutic target. So CD33 is a transmembrane protein expressed on myeloid cells specifically, including these myoblasts. The therapeutic potential lies on its limited expression on the hematopoietic stem cells and other progenitors, which reduces these sort of co or off-target effects. So for example, if we look into this gemtuzumab or zogamicin, it's an antibody drug conjugate that uses CD33 as a delivery mechanism for being able to look at sort of the potent cytotoxic agent. And it also induces DNA damage and the apoptosis of sort of acute myeloid leukemia cells. So this is quite an important mechanism that relies on the specific internalization of CD33. Okay. So I guess moving forward, why should CD33 be targeted? So CD33's expression in early myeloid progenitors makes it a prime candidate for targeting sort of these acute myeloid leukemia blasts. However, we've noticed from previous literature that the variability in CD33 expression among these myoblasts introduces a very sort of significant challenge, especially when we're trying to look at treatment uniformity. So CD33 is involved in inhibiting signaling pathways that regulate these phagocytosis and the immune response. So this could be something that could be sort of, I guess, understood further. And how can we really understand this further? So using the talk to data system, we are able to then query and sort of understand more how particular genes are expressed and in what sort of cell types we're seeing this expression. So in order to run this on the BioTrain system, you would go into the talk to data system directly, and you'll notice that we have, I guess, three different types of searches that you can run through the talk to data. So the first one would mainly be, in this case, the genes search. So this is where we can particularly add our gene of interest, which is CD33 in the context of sort of AML. And we see here that based on the cell ontology map of the CD33 search, we're able to then identify that there's a large number of cells that are expressing this particular marker, but we do see that most of the percentages of cells are within these myoblasts. So what does this tell us further? So we can see that these myoblasts in the early stages are in the myeloid cell development, and we can also see that CD33 is expressed very early during the hemoptytosis sort of lineage, which might explain sort of specific therapeutic target windows. So if we then click on the myoblast, we're able to see this distinction a bit clearer. So you can see that it's mainly sort of in the, I guess, earliest stages. And we can also visualize very particular studies within TalkTo data that are also expressing CD33, particularly in myoblasts. So we were able to really detect specific data sets with that information as well. Okay. So I'll go back. So how, I guess, we can take this further. So how can we sort of particularly focus only on myoblasts within acute myeloid leukemia, and how can we sort of increase the statistical power of these cells by integrating multiple data sets relating to that area? So in order to do that in TalkTo data, you can also remove any of the previous searches. And in this case, we'll be using the filter tab. So in order to be able to identify very particular studies in acute myeloid leukemia, we would need to use the standardized ontology system. So in this case, using the condition label, which then allows us to sort of identify very particular data sets relating to AML. So we can see here that there's about 39 different data sets, particularly focusing on this area. So we would be able to actually have more myeloblasts to be able to then understand the CD33 target expression further. So once we search for the condition, we're able to then identify all the different studies. So you can see that even from just one study where you're only working with like 3,000 cells or 4,000 cells, or even just 50,000 cells, we can now generate a search that integrates over 970,000 of cells relating to just acute myeloid leukemia. And this sort of allows us to be able to then increase the number of myoblasts that we can use to really understand, I guess, the significance of CD33 and any sort of off-target effects. So this sort of ability to integrate the studies would be sort of allowed using the create custom data set feature. So you can directly go into this functionality, and this would allow you to then integrate all of these multiple data sets relating to your labels that you've added from the ontology. And you can then add particularly what type of atlas you're generating from the TalkTo data system and which are the match cells that you'll be sort of generating from here. Okay, so once this is created, it'll take a few seconds to sort of generate an integrated object with all of the standardized ontology. So in the meantime, while that sort of runs, we do have sort of the prepared atlas here. Okay. So this is normally how the atlas would look once you integrated all the different objects. And in this case, the first sort of analysis that we want to identify, so we want to see how CD33 is expressed, particularly in myoblasts. And we can show you sort of this information going into sort of the bar plot visualization tool from the TalkTo data system. So what can we see from the CD33 expression bar plot from the atlas? So the bar plot shows that CD33 is most highly expressed in myoblasts. And at the molecular level, this does reflect that there's a role in CD33 in blocking sort of the differentiation signals in these cells. So this sort of keeps them in an immature proliferative state. And what does this mean, I guess, translationally, if we're trying to sort of look at its effect in therapies? So this could sort of suggest that the therapies targeting CD33 can specifically eliminate proliferating blasts from the analysis. So if there's any interest, I guess, from the team in being able to generate these type of visualization tools. So when you're sort of in the Atlas feature for this acute myeloid leukemia case study, you'll see that we have different visualization tools that we can use. So this is where I sort of generated the bar chart where I was looking particularly in the expression of CD33 on myoblasts. Okay. So how can we further understand sort of minimal expression or how CD33 is expressed across different myoblasts in these different data sets. So another result that we were able to also identify from the atlas, so we're able to see that the heat map shows minimal expression of CD33 across different types of hematopoietic stem cells, progenitor cells, and this suggests that there's a favorable therapeutic window, reducing the risk of this hematopoietic toxicity in the cells. And what does this sort of mean in the context of CD33? So CD33 negative hematopoietic stem cells retain their self-renewal capabilities, and this has been shown in previous research as well, which allows to really recover normal hematopoiesis after treatment. So this sort of shows a bit more of how CD33 can be targeted in the context of sort of acute myeloid leukemia and other minimal off-target effects. Okay. So why can this be an issue, I guess, when it comes to actually providing treatment? So if we go back into our atlas and we subcluster myoblasts to be able to really explore this question further. So under the sort of cell type subgroup label for your atlas, you'll notice that there's a lot of different metadata labels relating to all the information that you can use from this sort of integration of different studies. So if we're mainly focusing on myoblasts, you have to make sure that these myoblasts are subclustered so that it narrows down the cells that you'll be focusing on during sort of any downstream analysis. So once the myoblasts are subclustered, and sometimes in order to visualize the actual distribution or dimensionality of the cells a bit clearer, you can also sort of switch from a TSNE dimensionality to a UMAP. So normally the UMAP method allows to be able to see those change of neighbors a bit clearer on the visualization tool. Okay, so once we have this, we can then sort of try to validate the CD33 targeting. So we can query any number of genes on the talk to data sort of atlas, and we can see how CD33 is distributed across these different myoblasts from these 39 different data sets. So you can see we're not really focusing on just one study. We're trying to build this sort of cross-study analysis on a larger number of myoblasts. So this is quite a large number compared to what you would normally have in one study. So what can we see here when we query CD33 specifically? So we can see that not all of these myoblasts express CD33. And specifically, if I try to show you lasso, so we can see that even within this cluster, for example, we see very minimal CD33 expression, and you can try to zoom in to see this a bit clearer. Okay. Okay. And what else can this sort of tell us in terms of its application for AML? So this shows us that approximately 86.9% express sort of this antigen, and this variability can lead to resistance or incomplete targeting in a subset of patients. So it really depends on sort of the etiology and sort of the expression of this gene and how it can be targeted. So CD33 negative blasts may have an upregulated or an alternative survival pathway, which can be mediated by other co-target effects or other markers that can be co-targeted with CD33. And why I bring up this sort of co-targeting effect? So we do see that on previous research, going into some of the recent research, which tried to sort of understand potential co-targeting with CD33, we can see that CD123, which is IL-3RA, can be a potential co-target with CD33 in the context of AML treatment. And we can see that also based on this literature, approximately 87.8% of these acute myeloid leukemia cells express CD33, while the other subset, 9.4%, express IL-3RA. Without any sort of concomitant CD33 expression. So what does this suggest? So it suggests that nearly all AML patients could be targeted through either CD33 or IL-3RA. And in terms of the co-target effect, so if we sort of focus on sort of the co-targeting of these two antigens, this could overcome this CD33 negative resistance and improve patient outcomes when you have sort of the co-targeted cells identified. So how can we actually visualize this on talk to data, especially since we built sort of the custom atlas? So what we can do is then query L3RA here. And we can try to then identify within sort of across these studies. So I'll zoom out and I'll remove the UMAP. So we can identify specific cells that are co-expressing both of the genes at the same time. So this would be sort of the inner column. When you're sort of searching for more than one gene, you're able to see the co-expression sort of ranking system, and you can also color it by other counts. So it really depends if you want to identify myoblasts that have a negative expression and those that are positive. You can switch to a binary. So it'll give you at what stage these cells are expressing the gene or not, and you can label those cells as well. Or if you're looking more at the co-target effect, you can look at co-expression as well, which would allow us to identify these co-expressed cells within the data set. So if we go a bit more into sort of this analysis, we can also see that CD33 and L3RA are co-expressed within our huge sort of larger custom data set. And this was reported with the literature and consistent with those results. So more so, the yellow dots do represent these cells that are co-expressing the two antigens, and the blue dots represent cells that are only expressing L3RA. So this sort of finding validates what we previously observed from other literature, especially in regards to being able to sort of identify these co-expressed cells and these two antigens within sort of acute myeloid leukemia samples. So how can we take this sort of understanding further in terms of the pathways or functional pathways that have a relationship with both the cells that had a negative CD33 profile and those cells that had a sort of co-target or co-expression of these two antigens. So in order to be able to really understand the functional sort of relationship or pathways that could be enriched across these two groups, especially when you want to see how can you target the patient if they specifically have this sort of profile of like these CD33 negative cells. What we can do is then go back to our atlas and you can sort of reset so that you're able to see the cells again clear without the gene expression. And the next step would be then to perform differential gene expression. So what we would want to do is be able to compare those cells that are negative for CD33. And again, in order to do that, when you are working on the atlas, just make sure that when you're querying those specific genes and you want to label those populations, you can switch to the color by binary unit, and this will allow you to be able to label those cells that are positive and those that are negative for CD33. So you can actually create a new annotation for those populations, especially if you're going to use them for downstream analysis later on. Okay, so if we go into our differential gene expression, just so I can show you how you're able to map this on the functionality in Talk to Data, you can go to create a two-group comparison. And the best way to be able to layer different levels of metadata, so especially since we're going to include CD33 negative cells and we're going to include the cells that had the co-target effect of both antigens, we can map them using the composition feature, and this will allow us to then choose the categories that we've labeled. So I have prepared these earlier because sometimes it takes a few sort of time to be able to label them. So we have our myoblast subpopulation, and we also have the co-targeted cells as well. So what we will do is then generate the chart so we can directly see our CD33 negative population, so those cells that were not expressing CD33 at all. So this will be, for example, our first group. Okay, and our second group will be specifically within the CD33 positive identified cells. We're interested in only those subset cells that have the co-targeting effect. So cells that are co-expressing both antigens at the same time. Okay, so we're interested in sort of comparing what are the functional pathways that relate to these differences and how can this sort of give us more insights into the appropriate treatment for these patients. So I'll add this here, CD33 positive and the IL3RA co-target. Okay. So once you have sort of your groups identified and you've added them into the two-group comparison, just make sure that you add the title so that when you go back, you're able to identify what results are from that specific sort of study design. And also you can input specific genes that you want to identify, so specific targets that could be related to treatment response or some sort of treatment-related co-regulated genes, you can also add them onto the gene set list so that it only concentrates on those list of genes or markers, as well as what statistical test would be the most appropriate for the specific analysis that you're doing. Some which could be, if you have low number of cells, it might be best to run a t-test, or Venice, which would run on larger number of cells and would allow you to actually identify upregulated and downregulated genes as well, and the enrichment pathways from there. So this would be the setup for the differential gene expression, and just to show you how the results would look as well. So if we go into some of these previous results on these two groups that we're interested in, we're able to actually see the sort of result or the output from this analysis. So we can see all of the genes that are differentially expressed across our groups. We're also able to then run downstream analysis, which focuses on the pathways that are enriched in our co-targeted group. So our cells that have these two antigens, as well as the pathways that are specific to the CD33 negative profiling. So this would be where you can access the pathways for these two differentially expressed groups, or you can generate a heat map to be able to map this hierarchical clustering. Okay, so a bit to show you on how these results would look. So what could we find from the, I guess, the pathway analysis looking at these two different profiles of myoblasts? So we could see that in the fatty acid transporters, so specifically this pathway remains significant in the co-targeted group. So what does it suggest? It could mean that this particular myoblast profiling has a lipid sort of metabolism that could play a crucial role in the survival of CD33 and IL-3RA positive myoblasts. If we then look into the other sort of end of the spectrum within these two myoblast sort of groups, we can see that the IL-18 signaling pathway was found mainly in the CD33 negative group. So this pathway suggests that the immune modulation could be a critical factor for these myoblasts that have this negative CD33 expression, which potentially offers sort of some guidance into being able to then apply immune-based therapies for these specific sort of cells and in these patients that would have this profile. We could also see that in terms of the complement activation pathway, this also was significant in the CD33 negative group, which indicates its potential role in immune clearance. Okay, so I guess if we go into sort of the results, and if we want to then validate, okay, what studies can I find in talk to data that would have both the CD33 and the IL-3RA expression within these myoblasts, we're able to see that not many authors have really tried to understand sort of the difference between these two sort of profiles of these myoblasts. So we could only find 4 studies where authors have particularly tried to identify the CD33 positive or the IL-3RA positive myoblasts, but we're not able to see where they're co-targeting or they're looking at the co-expression of these two markers, which is quite important. So we were able to see that there's specific functional pathways that are specific to the co-targeted group, as opposed to negative CD33 myoblasts. Okay, so I guess as a summary point of all the session today, to get an idea what exactly we covered and what sort of insights we were able to gather from these features in Talk to Data and the custom app list. So we could see that CD33 is highly expressed in myoblasts. So we were able to identify that when we searched for the expression level of that gene, and we were able to map it in the cell ontology at the start of the session. We could also see that CD33 is minimally expressed in hematopoietic stem cells and other progenitor cells, which creates a favorable target window and gives some insights into how the treatment can be targeted specifically. We were also able to see that being able to identify myoblasts that have a co-targeted effect, like have the antigen expression of both CD33 and IL-3RA, could be a potential co-target effect in these therapies for acute myeloid leukemia. We also noticed that at the end of the session, when we tried to look at studies that were particularly trying to investigate the expression of these two very important sort of antigens in the context of sort of myeloblasts, there's very limited studies. Even using the Talk to Data system, which has over 150 million of cells, we could still see that there's only 4 studies that really tried to understand the significance of this co-target effect. And I guess the last part, which was one of the huge, I guess, results that we could gather, especially when we're able to label those cells specifically across 38 different data sets, we could see that the co-targeted effect of CD33 and L3RA in AML myoblasts specifically could disrupt sort of the key pathways that these cancer cells rely on, such as their metabolism and how they have an ability to rapidly grow with therapies that can become more effective in targeting these fatty acid transporters. And other results that we were able to also identify from these CD33 negative cells, which is something that most papers don't really concentrate on. We could see that these cells mainly respond to sort of immunotherapies, and we could see that they had sort of an enrichment of the complement activation pathway and also IL-18 signaling pathway. So this sort of gives us a new avenue in showing how these cells have sort of a weakness in their immune defense and how we can use this to sort of provide more immunomodulatory therapies. Okay. So I guess from this sort of end part of the session, we'll go into sort of the Q&A session next. And we'll try to sort of answer some of the questions that some users might have had during the session. So I'll go into the Q&A now. Okay, so I can see that there's some questions already. So how can I see or I see the unit of average rank when you query the CD33 expression? What does this mean? So I guess to show what this user is trying to sort of understand. So if we go back into when we were looking for the expression of CD33 in the atlas, so we'll go back to the gene query, and when we looked for the CD33 and tried to map it on the cell ontology, this is the question. So what does this average rank mean? So for each cell in each single cell data set, we rank all of its genes by the expression level. So since this is actually running it across the entire database, it's not on an individual sort of data set. The underlying rationale is that if gene A is expressed higher than gene B in one cell, gene A should be ranked higher than gene B. So no matter which technology that is used to measure the expression level, we can then have multiple genes with the same absolute expression sort of value. And this creates sort of a ranking system where we assign it as an average rank across these different data sets. So this is a specific method, and this really relies on the assumption that a cell cannot express more than 5,000 genes. So we basically converted to a rank into having the sort of end of the ranking as 5,000 because we don't normally see genes that would be sort of expressing more than 5,000 genes. So this is what the average rank is. It just means that when we curate the data in the system, we have to transform the gene expression value across the different data sets that is relevant to the study. Okay. I guess another question that a user had, so are there any associations between specific CD33 expression patterns and patient outcomes or disease subtypes? So I guess this is a great question. So previous studies show that patients with high levels of CD33 expression on AML blasts tend to have a better outcome when treated with CD33 targeted therapies due to their better cell susceptibility. However, it's important to note that the CD33 expression is it's not the only factor that influences AML. So there's other genetic mutations that could be sort of researched further to see their significant role. And a way to do that is when you are performing enrichment test analysis, you can do that on an atlas level as well. So you can actually choose specific enrichment pathways that are related to chemical or genetic perturbation, especially if you want to identify any mutations or any other, I guess, significance that would be changing the outcome for these AML patients. So this would be sort of an additional step if you want to identify this other sort of factors. Okay. Do you see the same expression pattern in other myoblasts associated with disease? So this is also an interesting question. So CD33 is also frequently expressed in cases of sort of myeloid dysplastic syndrome and chronic myeloid monocytic leukemia. However, we also notice that there's limited single cell RNA-seq studies that are specifically focusing on these diseases. So we would need sort of more data to be able to really understand the effect of CD33 expression when it comes to these other myeloid-related leukemia subtypes. Okay. So is there any other questions from the team or any scientists that have joined the session today relating to sort of the features or how to generate the atlas? If we are not able to answer specifically your question, or it's more related to another disease area, but you want to apply the same I guess, methodology or the same atlas creation, you can also reach to us at support and we can go through a specific one-to-one session on your specific case study using sort of the atlas feature in Talk to Data. Okay. And I believe you might have the support at bioturing.com sort of email from the chat, so you can reach out to us. And one thing I do want to mention before we end the session today is to remember that we're currently running these huge curation programs to be able to increase the power of the Talk to Data system. So we are sort of have a free curation program open for Visium HD data if you're working or you're interested in public data sets on this new sort of assay, or also RNA bulk data. So we have a new smart bulk app that specifically focuses on RNA bulk seek data, and you can also request any public data curation for that technology. Okay. So thank you everyone for joining the session today. And I'm looking forward to seeing you on our next session, which will give more details on what topic we'll be focusing on for that session. Okay. Thank you so much, everyone.